Hello guys, you're welcome back to Slick Academics, and in today's class we shall be discussing experiments on aerobic and anaerobic respiration. In today's experiment, we shall be demonstrating anaerobic respiration by fermentation in yeast, and we shall be needing a few materials such as glucose solution, yeast itself, liquid paraffin or cooking oil, a vacuum flask, but in this case a test tube can serve, and pipettes. Now, yeast and glucose solution are prepared with cool boiled water. This is to ensure that dissolved oxygen is driven out. Now, pour the glucose solution into a test tube and also add your yeast. Now, using a pipette, add your paraffin oil or cooking oil. This is to ensure that the oil covers and forms a layer at the topmost side, preventing oxygen from actually dissolving into the solution. Now, cover the test tube or the vacuum flask you use for the solution with a cork. In this cork, pass out a delivery tube from it into another container containing lime water. You can also decide to add the thermometer in order to measure the temperature changes. Also, a control experiment could be set up, and the control experiment will have everything as mentioned earlier apart from the yeast. After some time, the yeast solution in the flask will be seen to be bubbling. Bubbles of a gas will be escaping through the delivery tube into the container containing the lime water, which will eventually turn milky. This shows that carbon dioxide is the gas being released. But in the control experiments, no bubbles of gas will be seen, neither will the lime water in the flask turn milky. So, in conclusion, since oxygen was excluded from the setup and the bubbles of CO2 turned lime water milky, it shows that yeast respires anaerobically and that fermentation has taken place. Now to our second experiment. We shall be needing some materials such as beakers, delivery tubes, potted plants or germinating seeds, lime water, corks, soda lime, U-shaped tubes, or any container that could serve, then capillary tube, water, and dark cloth if you are using a growing plant. The essence of using the dark cloth is to ensure that the plant does not undergo photosynthesis during the process. Now to the experiment proper. There are four containers here. We have container A, B, C, and D. And these containers are connected using delivery tubes. Container A contains soda lime, container B has lime water, container C has germinating seeds, container D has lime water. Now, at the extremities of these delivery tubes, we connect suction pumps. Now, the suction pump is used to blow in air into the containers, starting from the container A. And when this air passes through the soda lime, CO2 is absorbed. And then it passes through container A and enters container B, which contains lime water. It shall be observed that no color change is observed here. It doesn't turn milky, meaning CO2 is absent. Then this gas further escapes and goes into container C, where we have the germinating seeds. And from there, we move on to container D, where we observe a change in the color of lime water from this color to milky color. This shows that oxygen was used up and then during respiration, and then CO2 was given up, because our formal observation in container B, where we have lime water, no color change was observed, but when it got to container D, there was a significant color change to milky color, meaning CO2 has been released, which turns lime water milky, and oxygen was used up during this process of aerobic respiration.